For some reason, I started the lecture again, and usually the lecture button goes away, but today I noticed it didn't. And I logged in and there was nobody there, and I said, geez, where is everybody? And it turned out I was in the wrong Zoom meeting. Are there any questions?
I did have a question.
Okay.
It was about the unknowns project. I wanted to know if we figure out what our bacteria is, will you confirm if we're correct, or should we just assume that we're right and write the rest of the project?
I will confirm whether you're correct or not after you submit your report, but I'm not going to confirm it until that time.
Got it. Thank you.
All right. So you have to write your report without my telling you whether or not your species is correct or not. Now, don't be worried about that. The grading of the unknown report, I think there's only 5 points, maybe it's 10 points for getting the correct species. Let me look that up. I think I've got it here on my own computer. Of course, my computer is a little slow here. I'm running Zoom, and the lecture is still compiling, still making the lecture file. All right, where is that? There we go. This isn't the best one because it's out of 100 points. I don't think it is. Boy, this is so slow I can't even control. There it is. Not finding it here. This is the grading section. Yeah, it's not on here for some reason. That's one other place I can check. Like I said, I think it's only 5 points for if you get the correct species, and if you get the incorrect species, it'll be marked down. Up to 5 points. If you're close, it'll be just marked down a few points.
Gotcha. Thank you.
Okay, is that good enough?
Yeah. That answers my question.
Right.
Thanks.
All right, so let me state, actually, I didn't do this, and that is I did do some grading this weekend, and the last lab, which would be lab 8, the first run of it has been graded, and the last worksheet has been graded. Are you seeing my screen? I don't think you are yet. No. All right, so let me go ahead and get to the grade book. So I know the last lecture worksheet has been graded, and the last lab has been graded. I do need to grade, and I'll try and do that before Wednesday, grade the one more quiz 2A. I'll try and get that graded before Wednesday. I'll try and work on that tonight. Although I doubt I'll finish tonight, but hopefully I will finish on Wednesday. I don't know if I got anything else graded, but those have been graded, so take a look at that. And where's the lab? I have lab 00, lab 01, lab 2, lab 3 graded the second time. I don't have lab 7 graded. I'm working on that. All right, any questions? All right, let me shut down the grade book. Then I'll share my screen. Probably don't need that. I'll leave it there. All right. Should be able to see my screen now. So today we're doing lab 9, identification of gram-negative organisms. We will have lab in the future, but it will just be like a 15-minute session on questions and answers. So I'll log in every day during the lab for 15 minutes to answer questions. And if there are no questions and nobody's there, after 15 minutes, I'll log off. And I will continue it if there's enough questions until the last of the questions is answered. But generally, I'll just be logging in for about 15 minutes to answer questions every day in the lab. From now on, after today, we don't have any formal labs. Any questions about any of that? All right, but you should be working on your unknown project. You also should be working on your infectious disease paper. You need to get your infectious disease paper done by Saturday, August 13th. I believe that is this Saturday. Any questions about any of this? All right, if not, I'm going to shut this down. And then we'll begin the lab. Lab 9, identification of gram-negative bacilli. There are some reading, not too much, on page 131, 132, page 272 to 276, that should be done before you begin this lab. We have some video clips in this lab. Upon successful completion of the lab, you should be able to interpret the results from the following biochemical tests. The Einvik reactions, and Einvik stand for I for indole test, M for methyl red test, V for Voges Proskure test, and then C for the citrate test. So Einvik are a whole bunch of tests, particularly used to identify Enterobacteriaceae, but they can be used to identify other species as well. We're going to talk about the carbohydrate fermentation test, and so you should be able to interpret results from it, the motility test, and the hydrogen sulfide production test. Any questions about any of that? Good. The program for establishing the lecture file just finished, so my computer will now work faster. Be able to explain how to interpret the TSI, that's triple sugar iron slant, which is a test, the SEMS deep, and then growth on McConkey's or HE hectone enteric agar plates. Each of these is a test that actually runs more than one test at one time, and then use biochemical analysis to solve case studies involving gram-negative bacterial infections. You will have case studies today in the lab. Members of the family Enterobacteriaceae are all small gram-negative non-spore forming bacilli. They are also facultative anaerobes. There are 53 genera in the family Enterobacteriaceae. 24 of the genera have been found to cause human infections. They're all glucose fermenting, oxidase negative organisms. Most of the genera shown above are microbiota that grow in the intestinal tract. Therefore, we call them coliforms. They're all opportunistic pathogens in that they can cause urinary tract infection, wound infections, pneumonia, septicemia. We can grow in burn wounds. There are some common genera that are responsible for causing opportunistic infections, including Escherichia, Enterobacter, Klebsiella, Citrobacter, and Serratia. But several members of these family are human pathogens like Salmonella, Shigella. I'm sure you know that. Klebsiella, Yersinia. Several genera are associated with gastroenteritis, causing diarrhea, stomach upsetness, including the genera Salmonella, Shigella, and E. coli serovar 015787. So this variety of E. coli is a human pathogen. The Enterobacteriaceae are similar to Pseudomonas, Pseudomonaceae, but this family, especially Pseudomonas aeruginosa, they're all similar to each other in that they're small gram-negative bacilli. However, the member of Pseudomonas do not ferment glucose. They're all oxidase positive. Pseudomonas aeruginosa is an opportunistic pathogen and a human pathogen, meaning a frank human pathogen in immunosuppressed individuals, burn patients, and trauma patients. Pseudomonas aeruginosa is also implicated as a healthcare acquired infection or a nosocomial infection. And it tends to grow in any environment that's liquid, moist, even dilute soap solutions. It's important for microbiologists to be able to identify and perform antibiotic susceptibility testing on gram-negative bacteria once you get a clinical specimen. In this lab, common biochemical tests will be discussed that are used to identify gram-negative bacilli. And here we're looking at some tests for gram-negative bacteria, but we're not going to talk about these. We will talk about certain of the tests. The indole test. These are organisms which produce the enzyme tryptophanase that converts the amino acid tryptophan into indole and pyruvic acid. After overnight incubation, you add Kovacs reagent, which reacts to indole to produce a red color at the surface. E. coli is positive for indole production. And here we see tryptophan with the enzyme tryptophanase will create indole, which when you add Kovacs reaction will be a red color. And this photograph, for some reason, is kind of more of a purple color, but a negative is a yellowish color, no red in it. The methyl red and Voges Proskure tests are two tests which are started together in one tube. You then separate the tube after you incubate it, and you take one part of the tube and test it for methyl red, and the other part of the tube you test it for a Voges Proskure, or the VP test. Both of these tests are looking at the end products that result from glycolysis. The methyl red test is looking for acid products. The VP test is looking for neutral products. So in glucose breakdown by glycolysis, if you make acid products in glycolysis, you can use the MR test to determine that acid has been produced. If a neutral product is made, which is is it acetone? No, acetone. Yeah, acetone. The neutral product is made, then the test will show you that it'll be red, and that would be a positive result that neutral products were made from glycolysis. Any question about any of that? All right, so for the MRVP broth, you inoculate it with your unknown bacteria. You then grow it for 2 to 5 days. After that point, you separate the tube into two parts, one for the methyl red portion, the other for the VP test. For the methyl red, you then add the dimethyl red to it. If it remains red, then it is positive for the methyl red test. If it turns yellow, then it is negative for the test. I think I got a picture of that. There we go, methyl red positive, all red, methyl red negative. It's not red, it's yellow. For VP test, you take that portion of the tube and you add Barrett's reagent A, Barrett's reagent B to it. You shake the tube to add oxygen. You let it sit and incubate for about 60 minutes. And after 60 minutes, you then look at it to see if acetone is made. If acetone is made, you see redness in the tube, usually at the top. So this is VP positive, and this is VP negative. Any question about any of that? The good news is with indole, MR, and VP, if any of them are positive, it'll be red. And if it's not red, it is negative. Citrate utilization. This test is used to determine whether a bacteria has the ability to use citrate, which is a molecule in the Krebs cycle. In the citrate test, the only carbon source available to be used by your organism is the molecule of citrate. If your organism does not change the color of the tube, which is green, and you have no growth on the tube, then it is citrate negative. If you have growth on the tube and the organism changes the color of the tube to blue, it is citrate positive. Any question about that? So if citrate is metabolized, nitrogen will be metabolized. You make nitrogen, you use nitrogen, you make ammonia, which is basic, and a basic will change the pH to a blue color of the tube. All right. The next test we're going to talk about is carbohydrate fermentation test, or the carbohydrate metabolism test. In this test, you have a tube with media in it and an inverted Durham tube also with media in it. And then you inoculate it with your bacteria, get the bacteria to grow no longer than 48 hours preferentially between, I think it's 18 hours and 24 hours, you end the test. And if it does not change color or changes color to a pinkish color from red, it is negative for the carbohydrate metabolism test. If it turns yellow, then acids are produced when the sugar is fermented, and that would be positive for the carbohydrate fermentation test. You can also look to see if gas is produced, and if there's gas and it's yellow, we call it AG, A for gas, A for acid, G for gas. If it's positive, we don't go plus, we say A for yellow in color, and then this would be negative. Each of the tubes would have one sugar in it that can be fermented, and you can have different sugars. Like you could be testing for glucose fermentation or lactose fermentation or mannose fermentation. There's several different sugars you can ferment. And they're all done the same. You're just looking to see if the sugar can be fermented. And if it can, it'll change it to yellow in color with or without gas. If it can't be fermented, it'll be reddish in color or pinkish. All right, any question about that? All right, let's talk about the motility medium. The motility medium is a media that is semi-solid. It has a low agar percentage, and so the bacteria can actually swim through it. I don't know, think of it as very thin jello. It is solid, but just barely solid. If you stab inoculate, then the bacteria in the motility agar stay growing only in the stab. This is a non-motile bacteria. Let me blow this up if I can. I don't seem to have the, maybe that's the problem. I don't have the link for blowing this up, so, well, I can do it this way and just ignore that. You'll notice that the agar here is clear over here. You can see through it. It's not entirely clear, it's translucent, but the point is you can see through it, the agar here. This agar you can't see through. It's kind of opaque, cloudy. It's cloudy because we stab inoculated, and then the bacteria moved away from the stab, making it cloudy throughout the tube. Any questions about that? And that would be a very motile organism. It doesn't have to be very motile. It can be slightly motile and still be motile. If it's slightly motile, it will grow from the stab. So here's our tube. Not a very good tube, but you get a good enough idea. There's our stab mark. Boy, that's not very straight. Got it at an angle here, but that's all right. So if it grows only in the stab, that's non-motile. But if it flares out from the stab, and usually it flares something like this. If it grows away from the stab, that is slightly motile. Any question about any of that? If it's totally motile, the bacteria will be swimming everywhere, and then it'll be cloudy throughout the tube. All right. The hydrogen sulfide production test tests the production of hydrogen sulfide. Some bacteria can make hydrogen sulfide gas during sulfur metabolism. We test for hydrogen sulfide production in either the triple sugar iron or TSI test, the sulfide indole motility test or the SEM test, or the hectone enteric agar or HE plates. Each of these 3 different tests can test the production of hydrogen sulfide. If hydrogen sulfide is produced, you'll see a black precipitate in the tube or on the plate. Citrobacter, Salmonella, and Proteus all produce hydrogen sulfide. So here, what is this? This looks like a SEM tube. And we stab inoculated it. And this is turning black. So this would be hydrogen sulfide positive. This would be hydrogen sulfide negative. Usually hydrogen sulfide is produced anaerobically. So you usually see the black near the bottom of the tube, not at the top of the tube. And this tube is mostly black, but occasionally you'll only see a little bit, and it'll be down at the bottom. Let me show you HE plate. Here we go. HE plate, if it produces hydrogen sulfide, you'll get black colonies. If you don't have hydrogen sulfide produced, they will not be black colonies. You can also do hydrogen sulfide production in the triple sugar iron tube, in which case the bottom of the tube will turn black. Usually the top of the tube, the slant, is not black. Like I said, that usually is because hydrogen sulfide is usually produced under aerobic conditions. And this is aerobic. All right, any questions about hydrogen sulfide production? We're going to also talk about the TSI or triple sugar iron slant. This tube has 3 different sugars in it: glucose, lactose, and sucrose. The sucrose and lactose are 1% sugar. The glucose is much less though. It has 1/10 the sugar of lactose and sucrose, or 0.1%. To inoculate the triple sugar iron tube, you first get your bacteria on a needle, and then you stab inoculate the tube with the bacteria. So you get the bacteria on the tip of this, and then you stab inoculate. You should come down at least 2/3 of the way. You don't want to come down to the very bottom because you'll damage the needle on the edge of the bottom, on the glass. But you should come down 2/3 of the way. This is only down about halfway from the bottom of the slant. And you should go down 2/3 because we want to look at the growth of the bacteria anaerobically as well as aerobically. And in the top portion here, it'll be growing aerobically. Definitely on the slant, it'll be aerobically. Once you stab inoculate, you then take the stab out and get bacteria on the tip of the slant, and then do the slant of the agar, put cells on the slant of the agar. You take the needle and make S-like motions on the slant. And that's to grow the bacteria aerobically on the slant and then anaerobically in the bottom of the stab. Any question about that? All right. To interpret the triple sugar iron is complex, and generally I'll only ask you simple questions about it. Like in this one, I would ask you something like, which of the tubes had fermentation of a sugar? And you'd say tube 4 is yellow, tube 3 had yellow on the bottom, and tube 2 is yellow, but it's hidden by the black. Wherever it's black, you can assume that it's yellow. And so this would be the fermentation of the sugars. Tube 1, no fermentation of the sugar because it didn't change to yellow. It's complex in its interpretation because so many tests are being ran. You are looking for fermentation of a sugar. If it's yellow, you have fermentation, I should say all yellow on both the slant and the butt of the tube. If it's all yellow, you have at least fermentation of the glucose sugar and fermentation of one other sugar, lactose or sucrose, you don't know which, or maybe both of them. So maybe all three of them are fermented. If it is, well, let's talk a little bit more about tube 4. This is happening, fermentation of the sugar, both aerobically and anaerobically. We have gas production, why the auger is pushed up. There's a big bubble of gas here. And then the auger is broken. That's another sign that gas is produced. This tube is not black, so there is no hydrogen sulfide gas produced. We do not know if lactose or sucrose is fermented, or both of them, but at least one of them is fermented because the tube is yellow everywhere. Okay, and if one of the sugars is fermented, you can assume glucose is always fermented because glucose is the base sugar. In fact, lactose and sucrose will be converted to glucose. So glucose is always fermented if one of the sugars is fermented. In this tube, we don't know if both lactose and sucrose are fermented, but they may be, or maybe just one of them is fermented, but at least one of them is fermented. On this tube, only glucose is fermented. We know this because the slant is red, the base is yellow. The base fermentation happens slower anaerobically than it does aerobically. And so the base is yellow, showing that fermentation occurred. If we looked at the tube earlier, the slant would have been yellow, but it's only fermenting the sugar glucose, which is at very low concentration in this tube. And aerobically, the glucose was fermented on the slant until it was used up. Then once it is used up, the bacteria start metabolizing protein that will produce ammonia, which is basic, and that will change the color of the slant from yellow to red. Any questions about any of that? So in this tube, only glucose is fermented. It was fermented on the slant while the glucose was available, but during the time of the test, all that glucose was used up. And then the bacteria use the protein, which make a base, and that will change the color from yellow to pinkish or reddish. And that's why we know in tube 3, only glucose is fermented. Any questions about any of that? Of course, it's not black, so there's no hydrogen sulfide being produced. And there's no bubble or breaking of the auger, so no gas is produced. In tube 2, only glucose is fermented. because the slant is red. It was yellow if we had looked at it earlier, but it was used all up, and then the protein was metabolized, making it a base, ammonia, and that changed the color from yellow to red. The butt is all black, and that's hiding the yellow. If we could see the yellow, there would be yellow here. You can always assume that if hydrogen sulfide is produced, then a sugar is fermented. So we're going to say this is yellow. Okay, so if it's black, you can assume it's yellow. So only glucose is fermented, no gas is produced, hydrogen sulfide is produced. In this tube, no sugars are metabolized. That's why it's red on both the slant and the butt. No gas is produced. No hydrogen sulfide is produced. Any questions about triple sugar iron? All right, sulfide indole motility test for the production of sulfide, hydrogen sulfide gas, test for the production of indole, and then test for the motility of the bacteria in the media. You stab, inoculate the tube. See if I can't blow this up. Let me blow it up this way. You stab, inoculate the bacteria, and if bacteria only grow in the stab, the bacteria is non-motile. So tubes 2 and 4 and probably 3 are non-motile. I think 3 is non-motile. Let me look down here. Tube through motility negative. Tube 4, here's the stab, and you see how we have growth right here and right here. So this is a motile. Tube 1 is motile, that's why it's black everywhere. So in the motility, you look on the stab, and if the bacteria is only in the stab, it's not motile. If it's growing away from the stab, it's motile. You then add Kovacs reagent to the tube, and if the top of the tube turns reddish or pinkish, these tubes are indole positive. Tube 5 and the control are indole negative. And then the final thing, you look and see if the bottom of the tube is black. If it is, hydrogen sulfide is produced. In tube 1, we have hydrogen sulfide being produced. All right, any questions about the sulfide indole motility test or the SIMS test? I can't grab that, so I'll just look at it. All right. We'll be talking just a little bit about MacConkey's and Hectone enteric agar. Both of these plates are selective for gram negatives. So only gram negatives tend to grow on MacConkey's and HE plates. So if there's growth, it's likely a gram negative. If there's no growth, it's likely to be a gram positive bacteria. If there is growth, you can then use both of these plates to determine whether lactose was fermented. On MacConkey's agar, if it turns pink, or kind of a purple pink, then the sugar lactose is metabolized, producing an acid, changing the color pink. If the sugar lactose is not fermented, it will not be pink. And it'll be the color of the bacteria, which in this case is white or off-white. For HE plates, if the sugar lactose is metabolized, it'll be a salmon color. It'll turn salmon color. If the sugar lactose is not metabolized, it will be a non-salmon color, generally off-white or white, but it'll depend on what color the bacteria is. Remember, on HE plates, you can look to see if the colony is black, in which case hydrogen sulfide was produced. Any questions about any of that? All right, I already talked about that. So let's talk about lab 9. You're not physically going to be doing any of these biochemical tests. However, you will be held responsible for knowing how to perform and interpret them. We have some video clips for you to watch. One on carbohydrate fermentation, that test. And remember, each tube has one sugar. You can test, in our test, you can test 6 different sugars. Okay, but you can test more. I don't know how many total you can test, but you can just test different carbohydrates in the tube. You can, we have a little video clip on the SIMS test, a little clip on the MR and VP tests, a little video clip on the TSI or triple sugar iron test, and a little video on the McConkey's auger test. You also need to view some of the website Microbuzz. Let me go ahead and open that. For the Microbuzz, you need to look at the information on the Microbuzz website. Just a minute. I don't, for some reason, I don't have, there's the mouse. Don't have access to the mouse to move it around. For Microbuzz, you need to go through these tests to fill in this worksheet. And you can get the information either from the lab manual or from the Microbuzz website. So on the Microbuzz website, on the left here, it'll be, and I think I state here which button to select. Click on the differential test. You only need to look at the test. You need to fill in the information for this worksheet. So if there's one, like the lipase test, you don't need to look at because we don't have anything on that test on this worksheet. Any questions about any of that? So view some of the Microbuzz worksheet, looking at the information on the test. For the test, you need to complete table 1, step 1 of worksheet for lab 9. You also have case studies to do, and you'll be pleased. I grabbed all the case studies and put it in the lab module here. Case study 1. We have some information about the patient here. Read it. And then we did the following tests. We brew the bacteria on a blood agar plate. And you can tell the hemolytic pattern here. And then we took the bacteria and grew it on a McConkey's plate. And you can see something's happening here, the color of the bacteria here. And then we did the gram stain. You can determine whether this is gram negative or gram positive. And then we did some rapid tests. We did the catalase test and the oxidase test. It's catalase positive, oxidase negative. And then we did the SIMS test shown here, the MR test shown here, the VP test shown here, the Simmons citrate test shown here, and triple sugar iron test shown here. So get your results for these tests and then identify the species that's causing case study 1 in this prison inmate with the wound infection. Any questions about that? Then read case study 2. Sorry, I don't know why there's not more space between here and there. Mikey has a pet turtle, and Mikey isn't good about washing his hands. About 2 weeks after getting the turtle, Mikey develops diarrhea, nausea, and fever. His pediatrician orders fecal specimen for routine culture. And here's some of the tests. We can see the bacteria with the arrow denoting colonies of interest on a McConkey's plate, and then HE plate, also growth of the bacteria, the arrow pointing out the colony of interest. And then we did some further tests. We have the Sims test here, the MR test here, the VP test here, the Simmons citrate test here, and then triple sugar iron test here. Okay, so read the results of these tests and then identify the bacteria responsible for causing Mikey's infection. Okay. And I'll tell you, if Mikey had washed his hands after touching the turtle, he wouldn't have gotten sick. All right, case study 3. This is a motorcycle accident victim. He's been on a respirator ventilator for several weeks. And as part of routine culture and sensitivity, they suction him, his mucus is collected in respiratory cheek, and then they test to see what bacteria he has. Bacteria growing on McConkey's plate, colonies, a gram stain. We did the rapid test, the catalase positive, oxidase negative. We then took isolated colonies and then set up these tests, the Sims test, the MR test, the VP test, the citrate test, and triple sugar iron. So identify the bacteria that's growing in case study 3. Case study 4, we have a 37-year-old male patient who's a burn patient. And after a week of treatment, several areas to begin to ooze. The patient has pain, redness, and swelling. And the physician orders a routine culture and sensitivity test. You can also look at the coloration. I'm going to have to blow this up, of the patient. And we can see that there's coloration of the infection. We have some cultures, oxidase test here, bacteria colonies growing on BAP here, and this isn't very clear, so you have to blow that up. Oh, we have black darkening of the auger. I don't know if I can shrink that now. What's the problem with this page? Move it over here, maybe. Oh, I can't move it. I don't think that'll do what I want to do. No. Well, you can barely see the gram stain here, but you can determine if that's gram negative or gram positive. On day 2, they did further tests. Here we have it grown on McConkey's agar. You'll note that the bacteria has a green pigment to it, right here and right there. We then did a glucose fermentation test, Simmons citrate test, and triple sugar iron test. I'm surprised they didn't do an oxidase test on that one. All right, case study 5. There was a family reunion. And after the family reunion, many family members began experiencing abdominal cramps, nausea, vomiting within 32 hours. Several ran a fever. They were tested. So the bacteria was isolated and grown on blood agar. And here you can see the bacteria growing on blood agar, and you can see hemolytic activity occurring. And then it was grown on McConkey's agar right here. The bacteria are catalase positive and oxidase negative. And then the following biochemical tests were run. Sims test here, MR test here, VP test here, citrate, and triple sugar iron test. So use these case studies to complete the worksheet for lab 9. Let's go to the worksheet. And then identify the species here. This is for recording the information for the different tests. Record the information for case study 1 here. And then here's the key, the table you're going to use for the key. So use this table to identify the bacteria for your case studies for this lab only. Do not use this key to identify your bacterial unknown or for any other lab. Okay, so this key is only set up for this lab. And if you have Enterobacteriaceae, Citrobacter furundi, you'll look at the oxidase test as negative, glucose test is positive, glucose fermentation, lactose fermentation is positive, triple sugar iron, A over A, hydrogen sulfide production positive, motility positive, indole test not done, methyl red positive, VP negative, citrate positive. So use this test to identify the bacteria causing each of these case studies, and then write down the species you believe is causing the infection in the case study. Any questions about any of that? So in the worksheet, you need to fill in the table, both the positive result and the negative result. For oxidase, we have the first result for positive filled in for you. The oxidase test turns blue if it's positive. And you need to fill in the table for everything else. Like I said, you will get this information either from the lab module or from the Microbuzz website for filling in the table. And then complete 2, filling in the blank, what reagent must be added to detect the presence of indole. So fill that in and then do that for each question. Note that some of the questions like D has 2 parts. Simmons citrate uses the pH indicator. So name the pH indicator here, and then turns blue under either acidic or basic conditions. You got to either bold one of them or write it off to the side here, acidic or basic. Okay, so some of them you have 2 answers for each question. All right, any questions about the lab? All right, if not, go ahead and get started on the lab. I will be here until 9 o'clock to answer questions. But when the last student logs off, I'll log off too. Okay, so if you have questions, ask before the last student is logged off, meaning ask early. All right, go ahead and get started on the lab. Let me stop the recording.